But I am going to talk about host genetics also, just like Randy, except looking at natural variation in host resistance to PRRS and PCV2, rather than variation created by gene editing. And very exciting results, of course, what Randy showed, and I think very, very promising for biomedical studies and also for the production industry. Of course, a long road ahead yet, and it may be the silver bullet, it may not be the silver bullet. So there's still a lot of opportunities to use this combined approach, which I think is still, in the end, is still needed in terms of eradication, biosecurity, vaccination, and host genetics. And so we'll be talking about host genetics in terms of natural variation, but of course now we can also add it into that variation that is created by gene editing, which is important to note is not the same as creating transgenics, but using gene editing to as another tool in this toolbox. Okay, and so with that in mind, as new technology developed in terms of genomics and being able to look at many genetic markers across the genome, in 2007, a group of us started the PRRS Host Genomics Consortium. You've heard, I'm sure you've heard about this before, and I actually gave a talk on this also last year. So some of the slides I'll be showing also will be similar to what you saw last year, but we've done a lot more additional analyses and got some additional interesting results to show you. So it's a very collaborative effort, and it started with a PHGC funded by the Pork Board and USDA, NIFA and ARS, but has grown into a very international collaboration that involves quite a number of universities from the US, Canada, funding from the US and Canada, and very strong industry component in terms of breeders and also pharmaceutical companies. And a very long list of collaborators, including the group from Bob's group at Kansas State University. So a lot of different projects going on. Of course, we all know that there's a reproductive component to PRRS as well as a grow-finish component. I'll be talking about the grow-finish component, but there have been a series of different studies, experimental infection studies that have been done looking at the grow-finish component, and I'll be talking about some of those. John Harding talked here last year about experimental infection of sows, of pregnant gilts that he has been working on, very interesting results. And then also we've been working on field studies with replacement gilts, and actually a former postdoc of mine, Nick Sarao, who is here in the audience, has a poster on that here at this meeting, so go and visit that. So the idea of these experimental studies, all conducted at Kansas State, is to bring pigs to groups of 200 commercial crossbred pigs to Kansas State, give them a 1-week acclimation, and then infect them with, in this case, the NVSL virus, which is also the same virus that Randy talked about in the CD163 pigs that were infected by that same virus. And then follow them for 42 days. So basically the protocol that Randy also talked about that was used for those pigs. A lot of data being collected, DNA collected for SNP genotyping, and over the trials, the first 2 generations of trials, as I call them, 15 trials total, looking at infections with NVSL and KS06. KS06 was the virus that was used in the first group of pigs that Randy talked about. But in total, over 2,300 pigs challenged. One of the lessons of this genetics types of work, in terms of looking for natural genetic variation, is that you need large numbers of pigs. And we've been able to get that through the excellent work done at Kansas State University. And they represent quite a broad variety of commercial pig genetics that is used in the industry in North America. And this just shows the first 2 principal components of these in terms of the SNP genotypes. And you can see very clear clustering of the different lines that came from the different industry organizations, the breeders. One of the things that is also done is to replicate some of these. So there are these pigs, for example. This cross was infected in one trial with NVSL and in another trial with KS06. So we can look at the ability to, for that same genetics, to how it responds to these different isolates. And they are quite different isolates, as we'll see. So the typical data we get, growth rates, most of them keep growing, not as fast as you would expect without challenge, and more variation. And this is the typical viremia curves we see, which are very similar to what Randy showed in the pigs that were not resistant to PRRS. So they all get infected, and here you can see there are no pigs that don't develop viremia. And unless they haven't sent me the data, but I haven't seen any of the data that was sent to us from Kansas State. There's no pig that doesn't develop viremia and doesn't develop antibodies. So likely resistant pigs, at least the ones that we've looked at, among the 3,000 we've looked at so far, there are no pigs that are completely resistant. We do see this rebound phase, which is primarily due to the virus escape rather than host genetics. And for that reason, we focus primarily on viremia area under the curve for the first 21 days, which we call viral load. And we found that to be fairly heritable, heritability of 41%, meaning that 41% of the variation you see here is due to differences in host genetics. For body weight under challenge or weight gain, it's about 30%. There is a relationship between viral load and weight gain. On average, pigs that have higher levels of viremia, they grow less fast, which is as you expect. But it's not a strong relationship, not at the phenotypic level. It's a little bit stronger at the genetic level. So this says that genetically, pigs that develop more, allow more viremia to develop, they grow slower genetically. Okay, so we've done a lot of studies doing genome-wide association studies. Those pigs were all genotyped using the 60K SNP panel, 60,000 genetic markers across the genome, and then we look one by one whether any of those SNPs are associated with differences in viremia and weight gain that we see. And fairly early on, we found what we call a major QTL, a quantitative trait locus. So a region at the end of chromosome 4 that had a major impact on both viremia and weight gain. These are the 3 genotypes at one of the SNPs in that region, which is we call it the WUR SNP or the WUR SNP. That captures most of the effect of that QTL. And you can see that the AA pigs, the pigs that have the AA genotype at that location, they have higher level of viremia than pigs that are AB or BB. We don't have a lot of BBs, but from what we've seen, the AB and BB are very similar in terms of what they show in phenotype. So that means that the B allele, the favorable allele, looks to be dominant. You only need one copy of them to get a favorable effect for that region. When we then look at weight gain, we saw that same relationship, but in the opposite direction. But again, the AA genotype being unfavorable, having lower weight gain than the AB pig or the BB pig. Now, these may not look like huge effects, but they do explain 10 to 15% of the genetic variation that we see in these populations. So that was the NVSL challenge trials. And for the KS06 challenge trials, for viremia, we see the same result. Okay, these 2 viruses are quite different in terms of virulence, and we see the same result. So that indicates that this QTL gives protection across isolates. However, for weight gain, the trend is still there between AA and AB, but it's not significant. The effects are not as large. One of the things you can see also here is that the level of viremia with this virus is lower, and the weight gains are higher. And that just indication that this virus, this strain of the virus is not as virulent, is not as hot as the NVSL. And it's the NVSL virus strain that was used with Randy's CD163 pigs to show that they were resistant. So work that done by one of my graduate students, Andrew Hess, who is in the audience here also, and actually will be defending his PhD next Wednesday. He looked at viremia curves for the NVSL infection and the KSO6 infection for animals that are AA versus AB. And you can see that the blue ones here they have lower viremia already fairly early on. And both for NVSL as well as KSO6. That difference is maintained in the NVSL infection, but not in the KSO6 infection. And here you can also see the difference in the levels of these curves between the two isolates. Now, these differences may seem small. It is on a log scale, but biologically they are relevant because they do translate into differences in growth rate. When we look at growth rate here, you can see that this subtle difference does translate in quite a difference in growth rate between the favorable genotype and the unfavorable genotype, difference of about 2.5 kilograms over 42 days. But for the KSO6, the difference is much smaller. Okay. Now, we have been able to identify what we think is the putative QTL, or the causative mutation. We've published that recently. It's in the GBP5 gene, which is a gene involved in innate immune response. That's been published. And there is actually a poster by Martine Schroyen, who is here also, a poster here that talks more about the difference between the AB and AA animals in terms of RNA-seq data that we've collected on these animals, so comparing AA versus AB. And based on those RNA-seq data, she has developed a hypothesis which suggests that, well, one thing we find is that the AA pigs develop a truncated GBP5 protein. The mutation it inserts an alternative splice site, and that results in an early stop codon. And so the AA pigs don't produce a complete GBP5 protein, and her hypothesis is that results in the PI3K AKT pathway not to be turned off, allowing continuation of viral entry and replication. And so go see her poster if you want to discuss that further and see more information on what's behind that. Now, I mentioned that these QTL, they only explain, they explain about 15% of the genetic variation, this one QTL, so there's still a lot of other genetic variation that is out there that controls response to PRRS in pigs. And so the question is, well, where is that other variation? And this is a GWAS, shows a Manhattan plot of, for the two viruses, based on all the data that we have. So the genome is along here. And then the level of significance that we obtained, the dotted line is a genome-wise significance level. The WR shows up very strongly for, so the chromosome 4 region, very strongly for both isolates. But for the rest of the genetic variation, we don't find any other large effects. There may be some hints here and there, but none of them that reach genome-wide significance. So it indicates that the rest of that genetic variation may be very polygenic, may be controlled by many genes across the genome. And the other interesting thing is that we don't see the peaks for the two viruses to be necessarily at the same location. Now, that can be just a result of lack of power, because when we estimate the genetic correlation between viral load in response to NVSL infection versus KSO6 infection, we find that genetic correlation is actually quite high. So indicating that genetically we expect pigs to respond similarly with the same genes regardless of whether infected with KSO6 or NVSL. And this is data or estimates obtained by Andrew Hess as part of his research work. Now, so the genomic regions don't overlap between isolates, but when we look at using GO term enrichment, we do find, and this is work done by Emily Wade, we do find that there are common GO term pathways. So it does look like there are the same pathways involved in protecting pigs against one isolate versus the other. Emily Wade is another PhD student, and she defended her PhD last month, working on this project as well as the skid pigs that we have developed together with Kansas State. For weight gain, the similar picture. So for NVSL, WR shows up very strongly, not so much for KSO6. But again, the rest of the genome, the rest of the genetic variation looks to be across the whole genome and fairly small effects. So then the question is, well, can you do anything with that? We know we can do something with the chromosome 4 region by genotyping animals for that SNP or that causative mutation and then select for that. But can we do anything with all these small effects? And the answer is yes. And using what is called genomic prediction, which is new methodology that has been developed over the past 10, 15 years and has been applied very effectively in dairy cattle breeding and is also being used now in swine breeding, selecting for traits like growth rate, litter size, and so on. But we think it can also be used to select for disease resistance, for traits for disease resistance that may not be affected by a limited number of genes, but could be very polygenic. And the idea is that you create a training population of pigs, which we have, where we have phenotypes for the traits we're interested in, and also the SNP genotypes, and so we have that in terms of the trials that we've done. And then use those to estimate the effects of not just the big effects, but all the effects, all SNPs, and using genomic prediction methodology, using some sophisticated statistical methods, getting effects not just for the significant ones, but for all SNPs across the genome. So every SNP that you genotype, you get an estimate of its effect, and it's properly treated statistically so that we don't, we deal with the uncertainty that is associated with those estimates. And then once we have those estimates, we can look at another group of pigs, get their SNP genotypes. We don't have to get their phenotypes. And then using the estimates of the SNP effects, get what is called a genomic prediction. So by just genotyping the animals, we can do genomic. We can get an accurate. We can get a prediction of how well genetically this pig is for response to PRRS. And this is the ideal situation for if this works. It's an ideal situation for dealing with PRRS, because breeding companies, they don't want to infect their nucleus herds with PRRS, obviously. So they need to get their phenotypes from outside. They could come from experimental trials. They could also come from the field, to create a training population and then estimate those SNP effects and then apply them to their nucleus animals that are genotyped for their same SNPs across the genome to get genomic predictions. Now, how well that works, you can evaluate by if you do have phenotypes on these animals, then you can correlate this genomic prediction with that phenotype, which we call validation. And that's what Emily Wade has done. She used either the NVSL trials for training or the KSO6 trials for training, and then looked at and then the other trials for validation. When she trains on NVSL to predict KSO6, she got pretty good accuracies of prediction accuracies. This is using the whole genome, so using all SNPs across the genome. This is the accuracy of prediction when she only used the SNPs on the chromosome 4 region, so this major QTL. And this is the accuracy if she used the rest of the genome excluding chromosome 4. So what this shows is that the whole genome does pretty well, but about half of that accuracy comes from the chromosome 4 QTL. When she trained on KSO6 and then validated in NVSL, accuracies on general were a little bit higher. But again, about half of that came from the chromosome 4 region. So it does say that this chromosome 4 region gives most of the predictive ability, but the rest of the genome can also help, even though those effects are all very small. So that's a methodology that can be used in the industry. We've also looked at antibody response, and Andrew and Ben Tribble from Kansas State has done all the assays in the group there. So this is measuring total PRRS antibody IgG at day 42. So we only have antibody at day 42. And when he does a GWAS on that, he finds the MHC genes in the MHC class 1 and 2 explain a very large percentage of the genetic variation. Which is as expected. He also found that when looking at SP ratio for different classes of viremia profiles, that the rebound pigs, the pigs that show a rebound, tend to have or have higher levels of antibody at 42 days. And that may reflect the higher immune response, putting more pressure on the virus and it resulting in escape of the virus, rebound. He's also looked at how does SP ratio at day 42 correlate with viremia and weight gain over the whole period. And the blue lines here show phenotypic correlations, and the red lines show genetic correlations estimated from the data. These are for NVSL, viremia, weight gain, and these are KSO6. I'll point you, so obviously it's a very dynamic system, and it shows that the relationship between immune response and what you see in terms of viremia and weight gain is a very dynamic system during infection. The interesting one, I think, is one of them at least is here, where the genetic correlation between antibody at day 42 and weight gain is negative initially and then turns positive. And that may reflect that pigs that put a lot of energy into mounting an immune response, resulting in high antibody at the production at the end, or that they sacrifice early gain because they put a lot of energy into the immune system. But they benefit from that in the end. Okay? And so in the end, having a high immune response results in better dealing with the infection and higher gain later on. So there's some very, very interesting results that have come out of that. Andrew has also looked at tonsil viremia. So in Joan Lunny's lab, together with Kansas State, they've also looked at viremia in tonsils collected at day 42. And he finds that the persistent animals, so animals that persistently have higher level of viremia during the whole period in blood, they tend to have higher levels of viremia in tonsil. And he sees that in the NVSL, the trend is there in KSO6, but not as strong. So we found that there are genetic effects. We can use those genetic effects to predict response to other isolates. But of course, one of the things we want to know is, well, how will this work in the field? How will this work in practice? And so we got a grant from NIFA, quite a number of institutions involved, looking at what does this mean when you go to more practical situations? And the practical situations we're looking at are twofold. One is to do experimental infections, but do co-infection with PRRS and PCV2, as well as looking at vaccination, and also then looking under field conditions, and I'll describe those trials. And so the questions that we raise are, well, this chromosome 4 QTL, as well as any other QTL that we may find, do they provide protection under more realistic conditions of co-infection and in the field? And also, does it have an impact on improved response to vaccination? So can we develop, what Bob always talks about, can we develop or select for vaccine-ready pigs, pigs that respond better, are better protected following vaccination? And then there are also outreach and education components associated with that. So the trials we're doing in that, or have been doing, we've done 4 experimental trials, again done at Kansas State, 4 groups of 200 commercial crossbred pigs again. Half of them are vaccinated as they enter with a modified live virus for PRRS, and then the other half are not vaccinated. And we've also, to increase power to look at the chromosome 4 region, we pre-select them, half of them to be AA, which we expect to be unfavorable, and half AB, which we expect to be favorable. And so they're co-infected with PRRS and PCV2, resulting in PCVAD. So the diagram for the trial is they come into Kansas State, half of the animals in one room, they're all vaccinated. And then 28 days later, pigs in both rooms are co-infected with PRRS and PCV2 and then followed for 42 days. The field trials, and I'll show you some results of the experimental trials. The field trials, also 200 pigs, groups of 200 pigs, clean pigs that are moved into health challenge barns, and then followed for all the way to market. And again, collecting a lot of samples and weights. And I'm not going to show you any data on this. This is still a lot of the assays still need to be completed, so I won't be able to show you any data on that. So but the co-infection trials, this is the typical thing we see, and these are the non-vaccinated pigs. So in terms of PRRS viremia, as expected, they don't show any PRRS viremia up until the point that they get co-infected, and then we see the typical viremia curve for PRRS. For PCV2B viremia, it's a little bit more messy. Not all pigs are clean at the start, so this is at the time of co-infection, and very variable. And body weight, the typical body weights that we see in pigs in the other trials. So those are the non-vaccinated pigs. I'm going to superimpose the vaccinated pigs now. They have viremia right after vaccination. It is a live virus, so it does infect the pig and replicate, but you can already see that the secondary response after co-infection, their viremia levels aren't as high as the non-infected pigs. In terms of their growth rates, I'll show you some estimates in a second, and they get PCV2 also, of course. So when we look at the data, the phenotypes we analyze is we look at the area under the curve, prior to co-infection, after vaccination, after co-infection, and the same for PCV2 viremia. This is after co-infection area under the curve, and then for average daily gain, both before and after co-infection. So looking at the WUR effect of the vaccinated animals in terms of viremia, the non-vaccinated have no PRRS viremia, we see that same positive effect of the AB pigs on lowering PRRS viremia. Of the vaccine virus, which again is different than the viruses we had used before. So that was interesting to see. And we see that same effect on growth rate, with the AB pigs having higher growth rate and no effect for the non-vaccinated pigs. After co-infection, the vaccinated pigs, the effect of WR is still there, but is not as large. And this may reflect that WR has a role in innate immune function rather than adaptive immune function. But the non-vaccinated pigs see it for the first time, and so there the effect is very obvious and about the same size as what we've seen previously. In terms of growth rate, and then the other thing is that the effect of vaccination is significant also. So the vaccinated pigs have lower level of PRRS viremia than the non-vaccinated pigs. So that's a beneficial effect of the vaccine. In terms of the effect on growth rate, the non-vaccinated pigs actually, and it's a bit surprising, we didn't see a growth effect. So they see it for the first time. And we expected to see an effect on growth, but we didn't. And we don't see it for the vaccinated pigs either. But there is a significant difference between the effect of vaccination in that the vaccine actually overall reduces growth rate. It increases growth rate right after co-infection, so there's some protection there, but then later on they have lower growth rate. And for PCV2, now WR in the PCV only experiments which were done by Dan Chabano at Nebraska, he never finds WR to have an effect, and we're seeing that also. But if they see PCV2 after they've been vaccinated with PRRS, we do see a significant effect of WR in that the favorable AB pigs have lower level of PCV2 viremia. So that may indicate that because the AB pigs prevent high levels of replication, that may also have an impact on subsequent the ability for PCV2 to replicate. Now, some of this work, especially the vaccine response, has been published in a paper by Megan Niederwerder, was a PhD student in Bob's lab, and additional work is being done by Janelle Dunkelberger. So she did all the WR effects, and she's also been looking at correlations between different phenotypes, so pre-PRRS viral load and PRRS viral load. So when we look at, for the same pigs, their viral load after infection is correlated to viral load prior to infection. So pigs that have high response to the vaccine also tend to have a higher response to the actual co-infection. It's not a strong correlation, but if you look genetically, which is here, it is a little bit higher, but it's still not all that high. But if we look at pre-vaccination response of the vaccinated pigs and post-vaccination response of the non-vaccinated pigs, so those are both cases where the pig sees the virus for the first time, that has a very high genetic correlation. Now, granted, these standard errors are very large also, so we can't draw too many conclusions, but it does support that, the role, the primary response would be the same in terms of vaccinated versus non-vaccinated pigs. And fairly quickly for PCV2, the genetic correlation between the PCV2 we see in vaccinated pigs and non-vaccinated pigs, very high, but large standard error. And for growth rate, they tend to be lower in terms of growth rate at the different stages and depending on whether they are vaccinated or not. And I think I'm almost done, Bob. So another poster shown by, in this seminar or in this conference by Janelle Dunkelberger, PhD student, doing a GWAS on these different traits, and she shows some interesting results in the MHC, the WR effect, and some of these effects on PCV2. I think some of these effects and this effect is similar to what PCV2 here, are similar to what Daniel Chubano has shown in the PCV2 only infections. So some of those results get replicated when we do the co-infection. So conclusions, the vaccination co-infection design doesn't seem to be a good model for PCV-AD. PRRS vaccination does result, at least this specific virus, or vaccine resulted in amplification of the symptoms and the effects of PCV-AD. The chromosome 4 QTL had a larger effect on PRRS viremia upon first exposure to the virus, either as a vaccine or actual infection, and that confirms its role in innate immunity. Interestingly, the chromosome 4 QTL also had an effect on PCV2 viremia, but only after vaccination with the PRRS modified live virus. It does look that whether the animal has been vaccinated or not, there's different genetics involved to some degree, and that does indicate that there may be opportunities to select for pigs that are vaccine ready.